I will show you in my presentation that we have developed the technologies where the round year production has been achieved for biofertilizer using specific cultures. Then, of course, these are non-polluting and environment friendly, and there is no net contribution to atmospheric CO2 levels because both all these are consuming CO2, making photosynthetes out of that, and then releasing oxygen. Of course, just to share with you that we have a national repository which is now named as Center for Conservation and Utilization of Blue-Green Algae. And this particular national repository is recognized by National Biodiversity Authority under the National Biodiversity Act 2002 of Government of India. And this particular facility was established in 1986. Incidentally, I also joined in 1986 as a scientist, and I have a bit of contribution in establishing this particular facility, then strengthening it, and establishment of labs, as well as later on, being in charge of this particular center. And we have about 800 odd unialgal cultures of especially of a cyanobacteria with us, and all these cultures, they are preserved with short-term methods, and all these cultures are basically characterized for morphophysiological as well as molecular approaches. And just to mention you that any of the researchers or any of the, I would say, research houses who require the cultures, they can approach our center, and there is a procedure to get the cultures as well as to deposit the cultures. Just to show you some of these cultures which are there in the germplasm, these are some heterosister cyanobacteria. You see here Anabina, Cytonema, Haplocyphon, Vestalopsis, the non-heterosistrous ones, the Formedium, the Calothrix, Oscillatoria, Lynchpia, all these are there. And then we also do maintain some microalgae with us, based on our, I would say, research interests. Well, the applications of microalgae, they span to all different fields. You talk about food, you talk about feed, biochemicals, biofuel, biofertilizers, biogas, fine chemicals, the restriction enzymes, lot many things have been reported and commercialized also. Bioremediation is another aspect, and then nutraceuticals. What I shall be talking mainly will be about biofertilizers, a little about biogas, and the bioremediation, which is basically the theme area of this particular colloquium. Well, talking about agriculture, of course, these blue-green algae or cyanobacteria, they have been developed as biofertilizers for rice-based cropping system. And the basic, I would say, quality or property of these organisms is that they fix atmospheric nitrogen and supply it to the plants. And the second property is that the rice field conditions, the flooded water conditions, are conducive to the growth and metabolic activity of these organisms, and so that they can easily grow there. You don't have to create any special conditions, and they provide cheap nitrogen in an eco-friendly manner. And nowadays, the use of this BJ biofertilizer or cyanobacterial biofertilizer in integrated nutrient management as well as organic farming is a recommended practice. But then that is what, as a science student or as a layman, one can understand that yes, they fix nitrogen and provide it to the plants. But besides providing nitrogen, they have many other advantages, many other benefits which they give, so that it becomes a kind of a economical and viable option to use blue-green algae as a biofertilizer for rice-based cropping system. One thing is that these are free living, so they would be, just growing there and multiplying and then dying there and contributing to all the metabolic activities, whether nitrogen fixation or I would say adding a lot of biomass to the soil. And then of course, as I told you, that they don't require much of the nutrients. They don't take away any nutrients from the soil. So it is a low cost technology. And then once they're adding a lot of biomass, you must have, if you have seen these organisms grow, or maybe if you have seen somewhere standing water and you see these organisms, they have a lot of mucilage, lot of, I would say, polysaccharides. So all those things are added to the soil and they improve the soil health, the soil aggregation capacity, the soil water holding capacity, the soil organic carbon. All these things are basically improved by their growth in the soil. And they also contribute indirectly, I would say, because since these organisms are autotrophic, photoautotrophic, and they grow there, they do not require much of the nutrients. They take CO2 and water and no, I would say multiply. But then all of this biomass, when they die, it is all added to the soil. And soil, is a storehouse of microorganisms. All heterotrophic microorganisms are also there. And this becomes food for heterotrophic microorganisms. And the population of those microorganisms also increases and they multiply in a much better way. And they also contribute to a lot of many metabolic activities of growth promotion as well as biocontrol and many other applications of those heterotrophic microorganisms which are there. Their population also increases, and all this has been seen under different experiments. These organisms also release growth promoting substances like gibberellins, indole acetic acid, vitamins, and all these things they help in better crop yield. Nitrogen contribution I have talked about. Once all these growth promoting substances are there, the soil health is improved, the soil fertility is improved. That also adds to increasing the grain yield. And of course, because these organisms, they will be growing on the surface, so they also do not allow the weeds to grow. So the farmers, they are also saving on any labor which is used for removing these weeds or use of weedicides. So all these things are basically helping in giving this kind of a viability, this particular technology. Uh, well, these are, of course, different ways. We have a farmer-oriented technology also. We have, I would say, a cottage production system also. All these things have been developed, and you have different prototypes to multiply these organisms or algae. You have static tanks, you have kacha pits, polythene lined pits. All these technologies are available and farmer can produce it at their own place. But then we looked into that, yes, once the farmer is producing themselves, there are some drawbacks. The organisms which are coming are, those organisms are really potential organisms, and many more things were there, environmental factors. And so we developed a new technology of ready to use BJ biofertilizer, which we developed as a semi-controlled indoor production. And this is a pilot scale facility for BJ biofertilizer, which is there at our center. And this is a process which has been developed. We grow these, I would say, organisms. It's a mixed culture in a specific kind of a medium, and once it's fully grown, we harvest it, and after harvesting we mix it with the carrier in a specific quantity, and then we dry it, and then we grind it to a powder. And this is the whole process. We have a culture in our lab, we grow it, upscale it in flasks, and then we inoculate in the production tanks as you see here. And then we have the fresh biomass and mix it with the carrier, and we regularly assess the quality at all these stages. And then we grind it, dry it, grind it, pack it, and then store it. And we have these packets. And this is a 500-gram packet, which is sufficient for 1 acre of rice growing area. This is a small data which shows that under Delhi conditions, what is the average harvest which we get in the months, because the summer months of April to June, the harvest takes about 7 to 9 days, and we get a good biomass. And then July to September, the harvesting days, the total days increases and so on. But then round the year, we get the production of this BGA, and then we can produce this biofertilizer. So based on this, the commercial houses have come up and then they have taken up this particular technology. These are the recommendations for its application. I will not go into the details. Uh, uh, this is normally applied 3 to 4 days after transplantation of rice. Of course, we have, as I been associated with this since so many years, we have carried out lot many demonstrations throughout the country. These are some of the demonstrations which we carried out near in the National Capital Territory region. And you can see here that based on the location, there is an increase in yield which varies from 2% to something like 20%. That is what how the farmer is gaining by use of the BGA biofertilizer. And at some places, there is also saving of chemical nitrogen in the form of urea or DAP, whatever the farmer is using. Then we have done different experimentations with different kind of inputs in different states of the country. You can see here, here it is along with the nitrogen, chemical nitrogen, because normally whenever we are using green algae biofertilizer, it is giving you a saving of or a contribution of 20 to 30 kilogram nitrogen per hectare. That means in urea you have 46% nitrogen. So you can say about 50 kilogram of urea is saved per hectare. So, but then because of the regular use of these chemical fertilizers, the use of these chemical inputs in the soil has increased. It has gone up to 160 kilogram nitrogen or so. So using only blue green algae may not sustain the productivity nowadays. So slowly and slowly we need to basically recover our soils or regenerate our soils. So here we have seen that by partly supplementing or replacing the chemical fertilizer, you can see you get better yields, as well as this is here you see that this is without any chemical fertilizer in different states, and you can see here the percentage increase in the yield. And the classical example of, you can say, PB is Punjab, because Punjab, they are using not many chemical inputs. The soil is really deteriorated. So that's why the use without chemical fertilizer, with the use of only BJ biofertilizer, you can see there is a very less increase. Whereas in states like Maharashtra, Madhya Pradesh, or UP, you have very better, I would say, production figures. Similarly, we have also seen the effect on farmers' income with the use of BGA, and there is an increase of about, what we have seen is 2,500 to 3,000 Indian rupees per hectare increase, which the farmer gets by use of this blue-green algae biofertilizer. We compare different cropping patterns, you can say, the organic farming, the integrated nutrient management, as well as chemical farming also. And what we could see here, that in the rice wheat cropping system, the use of blue-green algae and other organic inputs, the farmyard manure and green manuring by Sesbania, all these things put together, because organic farming will be amalgamation of all the existing resources which are there. So looking into that, you can see here that the use of blue-green algae is, it very well complements all other organic inputs also, and gives you a much better yield in case of organic farming as well as integrated nutrient management. And here you can see we looked into the grain quality as well as straw quality. And again, you can see here the organic produce as well as integrated nutrient management produce was giving a better protein content, as well as the P content in grain as well as straw, or the potassium content, as compared to the only chemical input, I would say, produce. Similarly, we looked into the micronutrients of these grains, and you can see here all these micronutrients of iron, zinc, manganese, copper, all these were much better in organic farming and integrated nutrient management. So they were conserved in a much better way as compared to chemical farming. Then, of course, we carried out an extensive survey in rice bowl states of India, that is Punjab, UP, and Haryana. That how basically, because we have licensed this particular technology to 6 firms, and they are producing this and then they are giving it to farmers. And we wanted to look into that, what is the effect of this BGA biofertilizer, how much it is adopted, and how it is behaving in the farmer's field. And you can see here that because since Punjab, you have again, the concept is a little different, and you have very progressive farmers. They are not much bothered about because chemical fertilizer is easily available, affordable. They are using it. So the percentage under BJ biofertilizer is less, whereas in UP and Haryana, there is a quite good percentage of farmers which are have adopted this particular technology. And it is the semi-medium and medium farmers who are really enthusiastic to take up this new technology, because they are the ones who are ready to take the, I would say, the risk, or look into the innovation which is happening, whereas the large farmers, they are not much bothered. And on the other hand, the small and marginal farmers, they are only, you can say, hand to mouth, and they really don't want to take up any risks. They are happy with the traditional things which they are doing. That is what our study has shown. Further, what we have seen is that how much reduction is there in the use of chemical fertilizers. And again, you see here the percent change is maximum be observed in UP, Uttar Pradesh farmers, followed by Haryana, and then Punjab, whether we talk about urea or DAP, or we talk about other inputs of no potash and zinc and other things. Of course, then we also looked into that how basically its use has given yield advantage or the income advantage. And you can see here the use of blue-green algae in all these states has given a better yield as well as better income, whereas the cost of cultivation has reduced because there is reduction in the use of chemical fertilizer. And this is some of the observations which the farmers have given us based on their visual observations of their crop. These are some of the, I would say, firms who have taken up this particular technology from us, and they are producing it and giving it to the farmers or selling it to the farmers. Of course, all these companies, they have, we studied all these things. They have different ways of commercializing or popularizing or distributing these products. And they have adopted different ways of really going to the farmer or to the end user. And this particular study we published in one of the journals of Journal of Applied Phycology, that how basically this particular rural technology, which was there about 40 years back, is now is a commercial success. Of course, we have, because we are an agricultural institute, and we are promoting all these organic inputs, and we also conduct regular trainings, and lot many trainings are conducted at farmers' place. On-site trainings are there where we advocate the farmers how to really produce it, how to use it, and then we regularly keep touch with them and get the feedback from them. The impact of technology, if you see here, you must be aware that rice is grown in about 43 million hectares in our country. And if this BGA biofertilizer is applied in about 25% of rice growing area of the country, it will give a saving of about 4.3 million tons of chemical fertilizer nitrogen saving. If we consider that it is contributing about 20 kilogram nitrogen per hectare. So much of chemical fertilizer nitrogen input will be, I would say, saved. And India is not self-sufficient in producing this chemical fertilizer. Lot of it is, I would say, imported. And the government is giving lot of subsidy on this urea. About 80% subsidy is given. So one can save lot of foreign exchequer by saving on this and using this BJ biofertilizer. And as if we talk about the farmers, the monetary gains to the farmer is that with the application of BJ biofertilizer, a farmer can save up to, I told you that one bag of urea can be saved per hectare. This is, of course, it is available about 480 rupees, but if we say 350 per hectare is saved on chemical fertilizer, and he gets a yield increase of about 10% or so, which gives him additional benefit of about 2000 or so. So this is the monetary benefit which the farmer gets. But then besides this, the other benefits, as I do, if you remember my slide on use of this BJ biofertilizer for sustaining soil health and other things. So slowly it is going to improve the soil and then make it sustainable. That is what is the aim, so that further inputs can be reduced. We have also developed now this BGA-based liquid formulation, and we have tested thoroughly its consistency as well as its physical behavior. All these things have been done, and last 4 years we are giving this biofertilizer to the farmers, and its shelf life is about 24 months, and you can see here it is maintaining its metabolic activity on storage at room temperature till 24 months. Statistically, it has been seen. And then we compared both the carrier based as well as the liquid formulation, and both are basically performing on par. And we got the farmer which have reported about 11 to 10 to 11% increase in the yield in about 50 odd demonstrations which we carry out every year. This is the scientific analysis which we did in our field at our institute, where you can see that how the available nitrogen and organic carbon values. This is a actually 4-year experiment where we have seen that the liquid fertilizer, when it is used, it gives the nitrogen availability increases by about, say, you can say 12 to 15% in different kind of treatments, taking the recommended dose of fertilizer full as well as 75%, because this is providing phosphorus also. This particular liquid formulation is also providing phosphorus because it has a p-solubilizing organism also put into this. And similarly, organic carbon also increases by about maximally 15%, where we are having full nitrogen dose and full potassium dose, but 50% of phosphorus dose, which is replaced with rock phosphate and along with liquid fertilizer. This is about available P and K. Again, the available P has been increased something like 30% in recommended dose. So what I would like to say that these both formulations, the carrier based as well as the liquid based, these are doing very good without fertilizer input also, and along with the fertilizer also. We have seen all the combinations. I have shown you earlier results of carrier based in different states also, where we have used without nitrogen input and along with the nitrogen input. And here also in these demonstrations and these trials, you see here all different combinations have been taken, and they have all resulted in very good availability of all these nutrients. And once these nutrients are available in abundance, the crop yield, crop health, all these things improves. Well, the second part which I am talking about is bioremediation and wastewater treatment. Of course, the microalgae, they have different mechanisms of treatment. You have a uptake, selective uptake is there, or accumulation, which would maybe absorption, so many things have been reported, or there is sometimes biodegradation of these pollutants also. And then normally wherever these polluted sites are there, you will see one or the other kind of algae which is there. And so they are also basically referred to as bioindicators of pollution. And normally they say that if microcystis is there, they say that the temple pollution, normally in the temple ponds you will find normally microcystis. So these organisms are identified as bioindicators for the kind of pollution which is there, or the kind of nutrients which are there. And all these, because these I would say wastewaters or polluted waters, they will have a lot of nutrients with them. So all those nutrients are easily harvested by these organisms, and they reclaim sewage, heavy metals, and radionuclide nuclide drug recovery. All these have been reported from microalgae. This is one study. I am not going into the details of this. What we saw that there is a you have a waste product in the steel industry which is called BOF, basic oxygen furnace, and months of these big heaps of this particular waste are there. And the only beauty is that this waste contains a lot of phosphorus, about more than 1%. What we could see that these cyanobacteria, they were able to very well colonize this a basic oxygen furnace. And what we could see that they could really take up the phosphorus out of this, and then other things they are taking from the solar system and from the atmosphere. And this particular biomass can be used for many other purposes. We also did a small study on Chlorella vulgaris, whether it can be used for chromium, I would say, remediation from ternary effluents. And what we could see, we took this ternary effluent, and what we saw that at the original concentration, in about 120 minutes or so, of course, about 10% of chromium was basically absorbed by the cells in the original concentration, which was originally 3976 ppm, which was quite high. But once it is diluted, and you can see here, if it is 1:50 diluted, about 96% of the chromium is removed by Chlorella vulgaris. Of course, this was a small study. We did not go further into this, but then we did a comprehensive study on taking a biorefinery approach, where we looked into mass production of the algal biomass using wastewater, and of course then looked into the metabolic activity as well as the remediation efficiency of these microalgae. The only thing which we looked into or we stressed upon is that we tried to isolate these organisms from the wastewater, and we took this agricultural wastewater from our institute only, and we isolated about 6 different organisms or microalgae from this, and 3 microalgae we took from our germplasm collection. And then we optimized the whole process, where we optimize the inoculation rate and as well as the, I would say, dilution of the wastewater, and observations, the regular observations were taken, and then we compared it with the regular or traditional BG11 media and also cultivation in wastewater, and then we screened them for phycoremediation potential also. And what we could see here, that you can see the metabolic characterization which we did, and you can see here the organism C1, which is basically a cholesterol species, and then C14, which is a for medium, and then another one is A, which is basically is not doing very well, but oxanochlorella species, which is there, giving a good protein content. So these are the ones which were giving better, I would say, metabolic characterization or good biomass as well as good lipid productivity. And C. One was also giving better lipid productivity to us. So looking into this, we also looked into the phycoremediation efficiency of these microalgae or these isolates. And what again we could see that C1, which is there, Cholesterella species, this is giving a very good reduction, about 80% reduction of nitrates, about more than 90% for phosphates, and more than 90% for TDS, around 90% for TSS, and similarly another one IC, which I referred, Desmodesmus species, which is giving a very good reduction in EC, then BOD and COD. And if I remember correctly, you can see that the initial concentration of BOD was something about nearly around 100 milligram or 95 96 milligram, and it reduced to about 17.5 milligram, whereas COD was again about 150 and it reduced to about 24 milligram. So such they were very efficient ones. And looking into this, we also carried out this, you can say, Pearson correlation analysis between different, I would say, metabolic and remediation factors. And this basically revealed a most significant correlation in almost all the parameters, whether the relation was positive or negative with each other. What we could see that pH was, however, not significantly but negatively correlated with nearly all parameters except dissolved oxygen, because the dissolved oxygen increased and pH also increased from you can say 7.6 to it went to 8.2, and dissolved oxygen was increasing from 2.5 milligram per liter to about 11.8 milligram, about 80% increase I could see. And then basically the DO exhibited a significantly negative correlation with almost all other parameters, whereas all other parameters, they were positively correlated with each other, whether we talk about EC, BOD, COD, all these nutrients of nitrate, phosphates, ammoniacal nitrogen, and TDS and TSS. And of course, the primary mechanism of this nutrient reduction, it comprises, you can say, improved metabolic pathways of nitrification and denitrification as well, including the production of, I could say, as you see, DO is increasing, so production of molecular oxygen by microalgae during this process, so indicating, you can say, the elevated levels of dissolved oxygen and decreasing the concentration of other parameters. Well, this is again important once we are looking into the applied aspects of bioremediation of wastewater, that how these what is the structural diversity of these microalgae or the seasonal variations which are existing in that particular water body. And so we analyze the diverse structural diversity of green algae as well as cyanobacteria, and these samples were collected in July in 2022 and January 2023. And you can see here that in July you can see the abundance of chlorellales is there, whereas it reduced in January, and we don't see any chloropicales in green algae in July, whereas they were found in the month of January. Similarly, in cyanobacteria, you can see the chlorococales, they were dominating in July, whereas you see the nostrocales, where they are dominating in January. So this is again very important to look into, that what kind of organisms they are there throughout the year, and whether they will be, and what is their abundance, and looking into I would say the species diversity. And you can see here the chlorella species, the chlorella variabilis, then chlorella sorokiniana, they are the abundant in July. And once you go down to January, then there is a again different species they appear, but of course they are also there. And this is again very important once we are selecting a particular microalgae or cyanobacteria for bioremediation, how it is going to survive or being metabolically active during the course of its bioremediation or wastewater treatment. That is important. We identified these organisms. As I told you, C1 is Cholesterella, C14 is Forminidium, and IC is Desmodesmus species. And we basically shortlisted these three because they were able to grow in 100% wastewater that we used and were metabolically active and were also having, I would say, remediation potential much better than other organisms. And then we produced this on a mass scale, and the conditions about 20 days cultivation period was there. Dilution, we did not do any dilution. It was 100% agricultural wastewater. Inoculation, it was 10%. And these are the isolates which we selected, and we took this biomass, and then this biomass we used for biogas production. And then we also took paddy straw. No, paddy straw is another, I would say, of course, I would not say it is as a waste material, but it is another commodity which is again causing lot of pollution because the farmers are burning this paddy straw after the harvest, and it creates lot of air pollution and smogging, as you see, especially in this part of the country during November and December. There is a lot of hue and cry about all these things. So we thought, why not use this carbon-rich substrate along with the nitrogen-rich substrate microalgae and take this co-digestion process for production of energy or gas. So we had different treatments where we took paddy straw alone, the algal biomass alone, and their combination. These are the four treatments which are there. Paddy straw alone, these were all based on the CN ratio because the digestion is basically very much, I would say, affected by CN ratio. If it is around 20, between 20 to 30, the biogas production is more. And what we could see here, that these four treatments we did, took, and then you can see here the in this co-digestion experiment, the T3 and T4. T3 is your paddy straw with algal biomass having a carbon nitrogen ratio of 28, and T4 is also paddy straw with double the algal biomass, and both were able to give much better biogas production and methane yield. And the methane yield was something around 62 to 60% in both these treatments, which we could see. And besides producing this biogas, it also gave a digestate, which was leftover material. And this digestate was, we analyzed this for nutritional value. And what we could see, if you see here, and we compared it with farmyard manure and other kind of manure which are available or used by farmers, and we could see that it is very much comparable, or I would say in quantities of nitrogen better than those manures. So what we could see in the treatment T3, about 3.3% nitrogen, 1.5% phosphorus, and 1.2% potash. So looking into its manurial potential, we carried out a pot experiment with rice, and we had all these treatments to see that whether this can be used as a manure. So on one side, you are treating the wastewater. On the other side, you are also having production of methane, and the digestate is also being utilized as a manure. So this was basically done, and then we did this particular experiment, and I looked into the plant parameters, the yield parameters, the soil parameters, and you can see here the plant chlorophyll, the carotenoid content in plants, the sugar content, the protein content. They were statistically significant in T5 treatment, and T5 treatment was 75% of recommended dose of fertilizers along with the digestate which was used. And you can see, or T2 is the recommended dose of fertilizer. So there's the chlorophyll content that shows the health of the plant, and you can see here the chlorophyll content is better in T5. Similarly, carotenoid content, of course, is better in recommended dose of fertilizer, but then equally good in T4 and T5. The sugar content, if you see, uh, again, you can see T5 is doing well. Similarly, effect on the nutrient status of plant. Just wait, it is going. So you can see here the macronutrients as well as the micronutrients. Well, again, you see here that T5 is much better, followed by T2. And in the micronutrients, which include iron, copper, manganese, and zinc, again, the T5, it is giving you better micronutrient content in plants, followed by the recommended dose of fertilizer. And if you talk about the treatment on yield parameters, again, you see that the grain yield was maximum in T5, which is recommended 75% of recommended dose of fertilizer along with the digestate. And then you have other treatments, the recommended dose of fertilizer, which is near to this. And these are different, I would say, figures. They indicate the DMRT ranking. Statistically, you can see that the digestate is giving better results. Similarly, looking into the soil activity, soil enzyme activity, which is basically indirectly is the activity of soil biological activity or the microbial activity. Again, you see here that the activity of phosphatases, the dehydrogenases, the FDases, all is giving you much better results when the digestate is used, replacing 25% of the chemical fertilizer. And this is the soil biochemical status. Again, the soil chlorophyll, the microbial biomass carbon. You can see here the microbial biomass carbon is again much better. Even it is statistically maximum as compared to the recommended dose of fertilizers. And similarly, you see here the soil polysaccharides. All these things are basically adding to the soil fertility status. This is the soil physicochemical properties. Again, the pH, electrical conductivity, available nitrogen, available P, available K, organic carbon. And you can see here that organic carbon in control 0.5, whereas in T5 treatment it is zero point 0.8. So basically, it is sustaining the soil fertility in a much better way when we are using this organic input or the digestate, which is containing paddy straw and algae digested together. Mm, This is the soil micronutrient status. So very comprehensively, we have studied all the, I would say, different factors and whatever is to be required to look into the plant and soil status or their sustainability by using these inputs. Well, then another biorefinery approach which we took was on wastewater phycoremediation and valorization of wastewater cultivated biomass. Earlier, we have looked into how this can be used for gas production as well as their use, the digestate being used for agriculture purpose. And here we have looked into that how this algal biomass can be used as a biofuel, biodiesel, or for nutraceutical purposes. So here again, we have taken two kind of wastewaters, the industrial wastewater and agricultural wastewater. In fact, this agricultural wastewater is different. This is basically the sewage wastewater which is being treated and then used in agriculture, and then the wastewater which is coming out has been taken. So, and then this industrial water has been taken from Mayapuri treatment plant. So we looked into the basically we characterize these wastewaters, and we again see whenever you the problem with the wastewater treatment is that it is not something you have a one organism and it can be used everywhere. For every kind of a different wastewater or every kind of a water body, you need to have your own optimization, you need to have your own organisms and all those things. So here again, we try to isolate the organisms. These are the organisms which we isolated from these wastewaters. And then we looked into their efficiency of bioremediating these wastewaters. And you can see here Chlorella sorokiniana, which was isolated for industrial wastewater, was doing very well for reducing the or remediating the wastewater, whether we talk about TSS, TDS, BOD, COD, total phosphorus, about you can say 88 to 95% reduction on all these aspects was there. Whereas once agricultural wastewater was considered, of all these organisms, the Graciela emersoni was giving much better remediation potential. Again, looked into other aspects of phosphate reduction, ammoniacal nitrogen, nitrite nitrogen, nitrate nitrogen, total nitrogen, all these factors, all these pollutants or these nutrients, they were very efficiently removed by Chlorella sorokiniana from industrial wastewater, whereas the Graciela emersoni, it I would say outclassed other organisms when agricultural wastewater was used and was remediated. So this is that how basically one has to really look for a special or a specific organism while remediating or going for phycoremediation. Then we did some metabolic engineering to really optimize the process of uh, higher biomass production, higher lipid production, as well as removal of nutrients. So we did a comprehensive study of no step-by-step taking single factor approach. Then we took this response surface methodology where we took the input parameters of NaCl, magnesium sulfate and incubation time. And then this basically was further fed into another, I would say, statistical application, which is again very much a kind of a hybrid evolutionary algorithm, which is artificial neural network combined with the multi-objective genetic algorithm. This was taking all these three input factors, and then once you have multi-objective output factors, then this kind of approach is being adopted. And these are the interaction plots for response surface methodology. And you can see here that the coefficient of regression, which basically tells you the fitness of the model, is all near to one, whether we talk about total nitrogen, total phosphorus removal, production of biomass or lipids. And further, this particular RSM, as I told you that we adopted this hybrid evolutionary algorithm, which we mentioned as ANN MOGA tool. This was developed to investigate the, you can say, optimal solution for input parameters for maximizing lipid production along with cell growth. In this has been done in Chlorella sorokiniana, MSP1, and along with the removal of, you can say, these total nitrogen and phosphorus. And what we could see that this metric, which was generated using sodium chloride, again from 1 millimolar to 40 millimolar, magnesium sulfate from 100 milligram to 800 milligram, and incubation time spanning from 4 to 20 days. And basically, what we could see, you can see here the specific growth rate under optimized conditions of AN and MOGA is quite superior to the one under control conditions. And specific growth rate was to the tune of 147.434. And if you see this lower table, you can see that the values of nitrogen, total phosphorus are quite reduced in the final after 20 days of incubation, and you can rather 18 days optimization for this AN and MOGA. What we could shortlist was 18 days, and then you can see the nitrogen and phosphorus, remaining phosphorus is very less under these optimized conditions, and we have more production of biomass as well as lipids. And then this particular basically model was further trained and tested
Excuse
and
me, sir.
validated. Pardon?
Excuse me, sir. Sorry, sir, we are running out of time
Just
actually.
maybe a few slides, just few
Yeah, sure,
slides.
sure, sure.
Okay. So this was further distributed into 70% training, 50% testing, and 50% validation. And all you again you see here that the R scale values are again near to one, and the model fitted very well in this. And we could get a better remediation potential as well as production of biomass and lipid. And then we carried out the fatty acid profiling of this under optimized conditions, and we could see better production of you can say the MUFA as well as PUFA as you see here. And there are a couple of fatty acids which were produced in abundance as compared to the control ones, which can be exploited for their use in nutraceuticals, pharmaceuticals, or otherwise for biofuel purpose, depending upon it all depends upon what kind of optimization you do for what purpose. We also did the metabolomic study of this using LCMS, and what we could see that all these different metabolites, they were reported under you can say optimized conditions, and they have different properties of I would say nutraceutical applications, pharmaceutical applications, anti-cancer, antioxidant, and different applications. And there are few which have not been reported so far, and some of the new metabolites which we could get here. Well, just to, of course, I have talked about all these things, but the only thing which I would say is wastewater is an appropriate nutrient reservoir for the production of microalgal biomass. And phycoremediation of wastewater lays the foundation for an economically viable, sustainable, and environmentally feasible technology within the framework of circular bioeconomy. However, wastewater dynamics often affect growth of microalgae. So there is a need to screen and select microalgae which could easily acclimatize in changing nutrient composition of wastewater as well as climatic factors. And of course, use of modeling as well as optimization, all these will have an edge to give you better process optimization, and which can be upscaled and then used for I would say in practical purposes. And similarly, of course, there is no doubt in the my earlier part of the lecture about BJ biofertilizer. That is a proven technology. We have developed this technology, and this particular technology has also been commercialized, and it is giving much, I would say, trying to meet the demand of the society because many of the commercial houses are now producing it. Well, that is all which I wanted to talk. But besides that, we have been working on pigments. We have done extensive work on phycobiliproteins. We have developed a very cheap and economical process, which is about what we have seen is 10 times cheaper than what is being sold in the market. And we have upscaled this process also. We have devised a specific medium for its production under raceways for production of phycobiliproteins. And the beauty is that globally, the phycobiliproteins are produced from spirulina. But then we have come out or identified some diazotrophic as well as some non-heterosystous cyanobacteria, which are comparable to spirulina in production. Then we have also developed these spirulina enriched functional foods, which have better nutritional quality in terms of protein, iron, zinc, other micronutrients. And these are also under, I would say, licensing process. Then we have also carried out a project on integrated production of advanced biofuels and biocommodities, where biodiesel production has been again optimized using again a very cheaper fertilizer-based medium, sufla, in a specific quantity, where Dunajala tertiolecta has been shortlisted for much better production of biodiesel. So besides this, other things are also there, but because I was mainly concentrating on agriculture and environment, so that is what I wanted to talk about. Thank you very much.
Thank you very much. Thank you very much, Dr. Pabbi. It was such an informative talk about the BGA biofertilizer and its application in the area of agriculture. And now we have few questions, Dr. Pabbi. We would like to discuss these questions online. So one of the question is from the Ritu Kundu. Is there any post-treatment required for the harvested biomass before using it as a fertilizer?